Okay, can everybody hear me okay? All right, super. Okay, well, thanks everyone for coming on this unseasonably nice day, actually. As Laura mentioned, we're sponsored by the Udall Center, which is a large grant from the National Institute of Health. So this is a tiny fraction of your federal tax dollars coming back to you. And we're very grateful for that support. And we're very pleased to partner today with the Parkinson's Foundation. Just to boost a little bit what Laura said about the in-hospital safety issue, it's never great to be in the hospital. It's definitely a little more perilous for patients with Parkinson's, so these kind of resources are really useful for patients. Strongly encourage you to take a look at that. We're going to talk about today is two things which are important in the treatment of Parkinson's patients and some research that we're involved in these areas. And this is drug treatment, which has been the mainstay for many years with L-Dopa, and Miriam Bonin is going to talk about exercise interventions. I think it's fair to say that here at Michigan, we are a major center for clinical research on Parkinson's disease, and this is part of our charge from NIH is to talk a little bit about the type of research activities that we're pursuing here. So I'm going to start talking about, the question here is, how does L-Dopa work? And I'll just tell you at the outset is, I don't have the answer to that question, and actually I don't think anybody else does either, for that matter. And this is a little surprising because we've been using L-Dopa as an effective therapy since the early 1970s. So over 50 years of work, and this has been the object of a lot of scientific investigation, but I think we still don't really have a satisfactory answer to this question. And, you know, as all of you know, and many of you have experienced personally the use of L-Dopa, the basis for this treatment is the fact that quite a few years ago in the late 1950s and the early 1960s, a number of investigators discovered that there was a deficit of the brain chemical dopamine. And this cartoon just shows a little bit of how that's supposed to function. Dopamine is made by nerve cells in the substantia nigra, that's the SNPC in this image, and it projects forward into the brain to other parts of the brain where it delivers that dopamine to other brain regions. And in Parkinson's patients, for reasons that we don't understand, those nerve cells that make dopamine actually deteriorate. And the big question for how does L-Dopa work is the brain is fundamentally, it's an information processing machine. So what kind of information is being conveyed by dopamine, and how does it work in the brain? Those are really sort of the big scientific questions, and those are also the practical questions for understanding this therapy. And you know, we also know this not just from what were the original investigations, which were studies of post-mortem brains, but we can study this in living patients, and this is with a method called PET imaging. And you can see here these very bright colors. These are the terminals of the dopamine-producing nerve cells. And you can see in the top part of that image, that's the pattern in normal subjects. And then below that, you can see what it looks like in Parkinson's patients. So it's clearly quite different. Now, how does L-Dopa work in sort of a very basic sense? You know, the basic idea is we're trying to replace dopamine. Now, we can't give dopamine successfully. You could give it orally, you could give it intravenously, but it doesn't get into the brain. On the other hand, if we give L-Dopa, if we give that orally or we give it IV, that actually does get into the brain, and then in the brain it gets actually converted into dopamine. And that's sort of the nuts and bolts of the very basic mechanical way of how it works. But that really doesn't tell you the answer of what does dopamine do in the brain? What kind of information is this signaling contributing? And on the right part of this image, these are a number of things that have been looked at in terms of what does dopamine signaling do. So reinforcement learning, this is a particular type of learning. There's clear evidence that dopamine is involved in that. Action selection. So how do you choose to do one act versus another? Habit formation. So as opposed to thinking through something and doing it, how do you just do it automatically? These are all things that have dopamine signaling has been implicated in. And the last two, which we're mainly going to talk about, are what are called vigor and motivation. And what's meant by these concepts. So the three important concepts to talk about, first is bradykinesia. This is the term we use for slowed movement in Parkinson's disease. And this is the feature of Parkinson's that responds best to L-Dopa therapy. And bradykinesia doesn't just mean slow movement. It also means that when you're moving, you actually have a progressive decline in speed or in this amplitude of movement. And everyone's seen this. If you actually do something simple like tapping your fingers like this, normally I can just keep going at the steady rate, and with the amplitude of my movements doesn't really change. But with many Parkinson's patients, particularly if they're untreated, even a simple thing like this will gradually slow down and the amplitude of the movements becomes smaller and smaller. Now, the way psychologists describe these type of things, they use the term vigor. And vigor is the speed, frequency, or amplitude or strength of actions. This is sort of the basic term that psychologists use to describe these type of phenomenon. And an interesting point is that the more vigorous, the faster you move, the more strength you exert, that's more costly in the sense of how much energy you have to expend. And in our day-to-day lives, in all activities, we need to proportion how much effort we put into any given action. You don't want to expend effort unnecessarily, and you need to make enough to accomplish what you want to do. So this is where the concept of motivation comes in. Motivation is the process of proportioning how much vigor, how much amplitude or strength of your action is to the perceived value of what the outcome of that action or what it's going to cost you. So this is what motivation means to psychologists. And what's come up over the last 20 years is the idea that dopamine signaling is specifically involved in this process of motivation. And this comes originally from observations made by patients with Parkinson's disease. And this is one of the classic ones from two neurologists in Boston in the early 1960s. This is before the introduction of levodopa. This is a story about a woman with advanced Parkinson's disease who's bedridden. There's a fire in her home. And under the stress of this potentially very threatening activity, she gets out of her bed, rescues her infant granddaughter from the crib, and runs out of the house. And all neurologists have stories like this. Now, this is kind of an anecdotal story, and you can see down in red here at the bottom, this is the text from their description of this episode. What they concluded correctly was that the pathways for voluntary control are still intact in this disease, even though the pathological process seriously interferes or blocks their normal performance. So the basic machinery for movement is okay, but there's something preventing you from doing that, using that machinery normally. Now, this is just a single anecdote, and there's a lot of these anecdotes that you can find neurologists will describe. But several years ago, a group in Italy took advantage of a tragedy in Italy to try and study this more systematically. And in 2009, there was an earthquake in central Italy, and it just demolished the town of L'Aquila. So several thousand people were rendered homeless. And these neurologists were following a number of very well-characterized patients with Parkinson's disease in L'Aquila, and they went and systematically interviewed them. And they found in individual after individual, this was the repeated story of under the stress of the earthquake, the homes collapsing, very severely impaired patients would actually be able to move much more normally. So this is a real phenomenon. And this suggests, in fact, that under sufficiently motivating situations, people with Parkinson's can move better. And so this is not just under the conditions of really dramatic motivating stimuli. This has become a topic of considerable interest to a number of scientists, and in particular, two neuroscientists, Reza Shadmir and Ala Ahmed, who are also engineers, and they've studied this type of phenomenon in very simple movements. So this is a complex slide, and I'm not going to go through all of it, but basically in the A part of the slide, they're asking people to make a simple movement of their arm. And they ask them to make it over different distances and with different speeds. So the faster you make the movement, the longer it has to be, that's more effort. So then what you can do is you can try and make a model of that. And the idea is that, if a faster movement is more effortful, and so you want to conserve that effort as much as possible to meet your goal. But on the other hand, if you move too slowly, then that's not going to work either. So there's an optimum point here. And so what they do is in this model, you can find the optimum point of what's the best trade-off between moving too quickly versus moving too slowly. And then you can manipulate the model. So if you increase the value of the action people will move somewhat more faster. If you make the action more difficult, the optimum point is slower. Now this is a model, but this is actually the way people behave in these type of experiments when these people do these type of tasks. So even for very simple movements, our brains are making this kind of cost-benefit calculation about how much effort do I have to put out, what's the value of the movement. This is exactly the type of process that psychologists refer to as motivation. And so this motivational modulation of vigor seems to be a basic function of our brain. And in a very influential experiment done by several neurologists at Columbia University a number of years ago, they studied these kind of movements in Parkinson's patients. So this is the simple movement. You start with a target. You start with a starting point. You move to a target. You can vary how far you have to move, how fast you have to move. You can make so varying the difficulty of the movement. And again here, looking at this on the right here, you can see the value down here is absolute acceleration. This is a measure of how fast you have to move and then how fast you have to slow down to get to the target. And what you can see is as the movement becomes more demanding, as it becomes more and more difficult to control with that absolute acceleration, it becomes harder and harder for individuals to perform normally. That's what this value is. Now the black circles here are normal individuals, but the squares here are patients with Parkinson's. So as movements become more difficult, it becomes more and more difficult for these individuals actually to accomplish them normally. And this is interpreted as indicating that there's not a problem, based on other experiments that they did, there's not a problem with really performing the movements themselves. It's actually that they don't seem to respond normally to the potential reward value. And on the right here, you can see this is actually a little bit plotting the same data the same way. The UPDRS motor score is a measure of Parkinson's intensity. And the higher the score, the more dramatic the Parkinsonism. And you can see there's a relationship between the difficulty people have with this task and how severe their Parkinsonism is. So again, this plus a lot of other data done in Parkinson's patients, normal controls, leads to the conclusion that the problem with Parkinsonism isn't really a movement problem, it's actually a problem of motivation. So how do you proportion the amount of effort you're getting to what you want to achieve? And this fits in very well with, when scientists see a convergence both of experimental data and of theory, they usually find this very convincing. This fits in very well with an elaborate theory that was developed by some neuroscientists based on the idea that produces the idea that dopamine signaling actually gives you a measure of how rewarding your world is. And that general signal of how rewarding your world is informs how fast you're going to move. So again, this is a very persuasive idea. I think this has really been the best idea about what actually constitutes bradykinesia, slowness of movement, Parkinson's disease, that's been produced really in the last 20 years. And I think many of our colleagues think this is the answer. And in fact, Dr. Shadmir and Ahmed have written a whole book about this topic called Vigor, which is really interesting. And those of you who have an engineering background and like to look at equations, this is a great book for you, perhaps not so much for the rest of us. Now, the problem with this experiment, the problem with this idea is that many of these experiments were done in Parkinson's patients, and they may ignore a basic feature of how L-Dopa works. And the fact is, when L-Dopa works, this was shown really about 50 years ago in some of the original work with L-Dopa treatment. It actually has two components. So there's what's called the short duration response, and that's the response I think many of you appreciate. You take your medication, half an hour to 45 minutes, you start to notice it kicking in and you're moving faster. And then after a couple of hours to 3 hours, your medicine, the effect will start to wear off. And that is tracking the amount of L-Dopa that's in your blood. But there's a second component of levodopa action, which is called the long duration response. And this is a component that kicks in over a period of days to weeks of chronic treatment. And you can see here in this cartoon here, this is an experiment done by Jay Nutt over 20 years ago at the University of Oregon. And the bottom trace here, you can see this is one of his patients who's never been treated before, gets IV, intravenous levodopa. Their motor function improves, and then as the levodopa washes out of their blood, it wears off. Now, this is the same individual in the same situation a year later after chronic treatment. And this individual has not taken any levodopa overnight. They get the IV levodopa, they get the improvement in movement. But what you can see is their baseline ability to move in the untreated, non-levodopa taking state has improved over that year. So this is the long duration response. And this is as much as 50 or 60% of the response to levodopa in early to moderate Parkinson's disease. So our idea was that, in fact, the prior experiments with motivation in Parkinson's had actually underestimated the role of motivation, and that this long duration response would be associated with an increased motivational effect. And we studied this in conjunction with two of my colleagues in psychology here, Taraz Lee, who's a cognitive neuroscientist, and James Brissenden, his postdoc, and a number of our patients who were kind enough to volunteer. These are all individuals who had not been previously treated. So what we did is to study them at baseline, before treatment, after a single effective dose of levodopa, after several weeks to months of treatment to induce the long duration response, after they're not taking their levodopa overnight, and then finally in their completely treated situation after taking their levodopa on the routine basis. And we used this similar type of task to what the neurologists at Columbia had used. This is a movement task. There's a signal. You have to move a joystick outside this circle, and then if you move accurately or properly, you get a reward, and you vary the reward. And you can see the reward here is predicted to be $20. You complete the task, you earn $20. Now, we don't actually pay people. They do get a set fee for participating in the experiment. This is a fictional reward, but it does. But in normal situation, when people get these rewards, they will react faster and move faster with the $20 as opposed to the $5. That's a motivational effect. So what happens? So first of all, do we see the patients respond to treatment the way we expect? And they do. So again, this is the UPDRS score here. This is a rating of Parkinson's disease. This is what our subjects were on average at baseline. This is after a single dose of levodopa, that's better. And this is after the long duration response. This is 2 or 3 months after treatment. And what you can see is there's a gradual improvement, and here's their completely treated state. And this is pretty much the same thing when we ask people to do a simple tapping task. So what happens to motivation? So this is the first thing at baseline, doing the task. You can see people are slow, and there's no effect of increasing the reward. And here's the short duration response, a single effective dose of levodopa. People are definitely moving faster, but no effect of the reward. And here's the long duration response, the effect of chronic treatment. Again, moving faster, but no effect of reward. And here in this combined state, when people are completely treated, you can see people, in fact, do get faster under the conditions of the reward. So this is a motivational effect, but we only see this here. We don't see it with the short duration or long duration effect. Now, my colleagues also thought of another thing you could do, which is to look at movement accuracy. And, in theory, people would start from the starting point and then go to make a straight line to cross to the circle, but in fact, people actually will make a bit of a curve, and the amount of deviation of that curve is a measure of how accurate the movement is. And one of the things that's interesting about accuracy of movement is that it's also responsive. There are motivational effects here. So typically, the faster you try to do something, the less accurate you are. But if you're motivated, if you're getting an appropriate reward, you'll actually compensate, and you can reduce the inaccuracy of your movements under sufficiently motivating conditions. So this is the same thing here. This is the prior graph. Now we have movement error here. And you can see, in fact, people are moving slowly. As they try to move a little faster, there's no effect of the reward there. Same thing with the short duration response. Here people move significantly faster, but again, no effect of the reward. And here again, people are moving faster, more accurately, but in this case, they actually have a motivational effect. So this is a combined, putting both accuracy and movement speed together. You can see there's a gradual improvement, and these look like the results we get with the clinical measures of movement speed. So what do we know? So do we see this kind of motivation effect with the long duration response, this large chronic effect of treatment? Well, the answer is no, we don't. And what does this tell us? it tells us, in fact, I think that at least in this simple sort of experiment, that bradykinesia is not deficient problem with motivation. So this is quite different than what most people have concluded over the last 20 years. And that this reason I think that people have concluded this is because the experiment hasn't been done the right way to sort of take apart how people respond to levodopa. So we don't really know how levodopa works in Parkinson's disease, and this is something we're trying to do some more experiments on to see if we could sort this out. If we could identify this in a more meaningfully scientific way, it might be possible to actually improve therapy. So happy to take any questions. Yeah.
How does adrenaline fit in with the motivation?
Oh, so that's a really good question. Yeah, so I think the question is, how does adrenaline fit in with motivation? So first of all, if you look at the brain pathways that use adrenaline, they are not normal in many Parkinson's patients. It's actually noradrenaline, norepinephrine is closely related, is the more relevant one in the brain as opposed to in your body. They're not normal in many patients. We tend to think of that as being more involved in sort of alertness and attention as opposed to this type of motivation thing. But unfortunately, we have very good tools to measure in people. We can measure dopamine signaling with PET imaging. We can measure some other things with PET imaging. But no one has ever come up with a good method to actually measure, in living people, to measure norepinephrine in the human brain. So it's an unresolved question, and it's an excellent question. Mm-hmm.
What about the impact of sleep deprivation?
Mm-hmm. Yeah, that's the question. So what about the effect of sleep deprivation on motivation? Sleep deprivation has a lot of different effects. You know, it affects learning, it affects memory function, definitely affects attentional function. I don't think anyone has specifically studied this type of motivation phenomenon. There has been some recent work that this sort of idea, this motivation concept really comes out of interest in movement primarily, but recently some people have expanded this to look at what's called cognitive effort. So how hard do you have to think about something to accomplish something? And that possibly would be, I think, a more relevant avenue of investigation. But I mean, this is actually quite recent work, I would say, on this cognitive effort. Any other questions? Okay. Somebody holding up their finger over there?
Let's see.
Yeah.
What about measurement error? Is it getting in trouble right now?
Measurement error in these experiments? Yeah, that's a great question. So it's not, we're interested in accuracy because we think that's a component of this, and there's really two components of that, I would say. So you're interested in how do you actually measure. It turns out measuring accuracy of movement, even with simple movements, isn't a straightforward thing, and this is something that the psychology experts in this area disagree about. The method that we use, this sort of deviation from a straight line, is actually, I think, a reasonable metric. But there are, in fact, some other ways to do this, which might actually be better. So, yeah. Okay.
Thank you so much,
Oh,
Dr.
one more.
Allen.
We have one more question here. Yes, ma'am.
Just curious, how does stress impact or correlate with movement?
Well, I think stress in particular, I think the biggest problem with stress is probably related to attentional function. And, it's commonly used as executive function. So things that are distracting are more difficult to manage. And I think in particular, we talked mainly about movement today, but one of the other functions of the target region that receives the dopamine, the striatum, one of its major functions is actually participates in what's executive function, so actually switching from one task to another. And so under distracting conditions, I think this becomes a particular problem for people is maintaining their focus and attention. And something I hear from a lot of patients, even patients with very early Parkinson's disease, it's very difficult for them to multitask. I think you really have to focus just on one thing at a time and break things down into simple units. Okay, all right, excellent. Miriam?
Thank you, Dr. Alvin.